Morning students, today we discuss about pathogen monitoring by heterotrophic plate count. So, pollution is one of the most serious problem in our country. Then, especially for water pollution. So, water pollution is the contamination of water. Sometimes unwanted waters are used for the drinking, cooking, cleaning, and swimming and other activities. So, the pollutants waters contains the chemicals and the trash, bacteria, and parasites. All forms of pollution eventually make their way to water. So, how to detect them? Uh, Introduction, indicator of microorganisms. So the indicator of microorganisms means the E. coli or coliforms group of bacteria present in the pollution water. They are zones, types of waterborne pathogens which can be spread in drinking water. The goal of water treatment plant operator is to ensure that none of these pathogens are present in the finished water to infect the consumers. So the waterborne pathogens is too time consuming and expensive to be feasible. So the indicator microorganisms, presence of one or few which are usually present when pathogens are present. So these coliform bacteria, E. coli, salmonella, then some viruses are present in the waterborne. Then masses of coliform bacteria are discharged from the intestinal tract. With these animal speakers, if sewage enters the waters and the coliform bacteria in the sewage will be carried out along and will survive in the water for a long period of time to the possible presence of pathogenic microbes to contaminate for the or create the illness of the humans or uh, humans and animals, etc. So this image indicates the sewage contaminates the river or bond or sewage or otherwise sea. The total coliform of bacteria testers detected. How to detect the pathogens, especially in coliform bacteria. So the number of coliform bacteria present in a specific volume of water is a measurement of the amount of sewage which has been discharged into the water, and the large number of coliform bacteria are present, then a large amount of sewage has entered the water. So the water is probably unused of unsafe for drinking or swimming. A smaller number of coliform bacteria indicates a lower concentration of sewage pollution. Then characteristics of indicators microorganisms. So the good indicator microorganisms has qualities which make it easy to test for the lab. In general, indicator microorganisms are greater numbers are present than the pathogens they indicate. Lab then they are pathogens since the indicator cause no or only mild illness. So the coliform bacteria are good indicators because they only reproduce in the intestines of animals. So they will not be present in water unless the water has been contaminated with sewage. Then the coliform bacteria are also able to survive outside animals' intestines and will be present in water for several days after the water has been contaminated. So the total coliform bacteria present in the sewage and fecal contamination of water pollutions, mixing the water pollution. Then the most operators will deal with only two types of indicators. First one, total coliform bacteria and fecal coliform bacteria. Then how to test the coliform bacteria? So the water treatment plant operators can use test for indicator microorganisms in order to detect pollution to prevent the development of a major problem such as a disease outbreak. Then can also help the test operator determine the source of contamination to maintain the proper sampling and testing to operate the coliform bacteria, and mentioned previously test for indicator microorganisms help determine the efficiency of water and wastewater treatment plants. And also coliform bacteria are an integral part of operative reporting. Next, heterotrophic plating count to detect the coliform bacteria. The heterotrophic plate count methods, otherwise called as HPC, then otherwise known as standard plate count method. It is requiring organic components of carbon and nitrogen for nourishment in water. The heterotrophic bacteria only utilize for the carbon sources and nitrogen sources. So this type of test to detect the coliforms bacteria. This test can provide useful information about the water quality and the supporting data on the significance of coliform test results. So high concentration of the general bacterial population may hinder the recovery of coliforms. Then the HPC plate count is a microbial method that uses a colony formation on culture media to approximate the levels of phototrophic flora. Most bacteria associated with drinking water systems are heterotrophic. These heterotrophic bacteria are only utilize the carbon sources and the nitrogen sources. However, given indication of the types of organisms present on their sources. For example, that only 1% of total bacteria found using a direct microscopy and enumerated using HPC procedures. HP means heterotrophic plate count methods. In possible explanations of the difference include the presence of some bacteria and the culturable state and the fact that the HPC media do not provide the complex nutritional requirements necessary for the growth of all heterotrophs. The three different methods for performing the heterotrophic plate count method using the three different type of media will be described. First one spread plate method, second one core plate method and membrane filtrate methods. So which media and methods are used in the coliform pathogens detection? The three types of different media can be used to enumerate HP method, including the standard methods are it's shortly called as SMA and R2A agar and MHP agar. And all three these of media are non-selective in that they supply nutrients and support growth for a variety of heterotrophic bacteria. First one standard method agar, R2A agar. This method this agar used for the four plate technique and spread plate technique. Then another one is MHPC agar. This media also used for the membrane filtration method to detect the coliforms bacteria. These are one of the examples of membrane filtration method, spread plate method and core plate method. First one core plate method. Core plate method already we all know that this core plate technique can be used for any type of liquefied sample, the enumeration of bacteria. So give your conditions vary depending on their types of their bacteria being enumerated. Then this standard recipe held at 42 to 40 degrees Celsius in a water bath, and then 32 this dilution for all dilution were core plate technique for used. 32 300 colonies they were detected. 1.0 ml of the sample or dilution is transferred to the sterile every 50 days, and then agar is poured, then core plate method is finished. Then in the standard method agar 35 degrees Celsius to 48 degrees Celsius were used for the incubation period. Then finally the next day after 24 hours the result were enumerated or man-made or manually or mechanically. Then one spread plate method. Spread plate method is otherwise called as SMA agar. SMA agar standard methods agar used routinely for this spread plate technique to enumerate phototrophic bacteria. H2A agar can also be used. So the plates are allowed to warm to room temperature and dry before the inoculating. 30 to 300 colonies were observed in this particular plate technique for the all dilution. The standard method agar 35 degrees Celsius to 48 degrees Celsius for the incubation time. Totally 5 to 7 days incubation, and after the colonies were enumerated mechanically or manually report or mechanically or manually enumerate this type of plate technique. Thank you.